It's a pleasure to be here. I want to thank the organizing committee for bestowing this honor upon me. I'm not normally somebody who seeks out these things, but it's an incredible honor, and I'm very flattered. It's also great to be back at this meeting. Some of you have commented after having attended this meeting regularly in early 2000s. Since 2008, this is only my second time here. There are a couple reasons for that that I'll go through in the talk. So, of course, it needs to be said that all of us are building upon, the findings of others, and this is, I think, one of my favorite quotes, and a way to make sure we all remain humble, and that we all recognize that without the contributions of those who came before us, we would not be where we are. And of course, having role models is incredibly important. So I'm going to talk first about, as Jeff Lipton did, with some personal inspirations. I'm going to start with my mother's father. He was a family physician in a small town, relatively small city in India. To become a physician, he actually would read textbooks under the street light at night because he didn't have electricity, didn't have light to read within his home. And this is a picture of him on the left. And with his wife, my grandmother, on the right. Those of you who know a little bit about Indian culture will maybe recognize that he's Parsi or Zoroastrian. It's a relatively small religious group within India. His daughter, my mother, was named Alu Modi. This is a picture of her. She is probably one of the most kind, gentle, and just loving people that certainly I've ever met, and that maybe those who have met her will say the same. She's 92 years old. She lives very close to us right now. She's a little too old for long distance trips like this, unfortunately, but hopefully she'll get a chance to watch the video. So she was trained as a nurse, and on my father's side, my father's father was the first in the family to become a physician. The family at that time was living in a small village, and if it wasn't for him, the family would probably still be in a small village in India. And he became a tropical medicine specialist in large part because he took advantage of an opportunity of he's shown here on the right, of joining the man on the left, who was also a physician but going to train in England. And so the man on the left had some health issues, and they were worried about sending him alone, so they paid for my grandfather to go, and that gave him a chance to train in England, which was transformative because when he came back to India, he was basically an expert, and that completely transformed, my father's side of the family, his education and his persistence. My father was a cardiologist, not only a fellow of the American College of Cardiology, but a master, which is an honor that's bestowed upon a relatively small number. He's a pioneer in echocardiography. He is also 92 years old. He's shown here with his wife Iris on the left, no relation to the Iris trial, and on the right, he's been traveling through Europe in the last few weeks. So that picture on the right was taken earlier this month in Lyon, and he's here in the audience today. So it's really wonderful to have him here as well. So collectively, these people embodied several great qualities, which I've listed here. I've done my best to try to absorb as many of these can, some better than others. One that I want to highlight is towards the end, and that's support for women in both educational and professional opportunities. That's been really important on both sides of my family, and that was, I think, ingrained in me as well. So going back, high school graduation, my father and I, when we were younger, and just to prove that I had facial hair before Jerry, maybe, he told me at that time that my long hair was just a passing phase, and for most of the next 35 years, he was correct. But he would have been more prescient if he had just said, "Your hair is just a passing phase." But I'll give you credit. Okay. So with that, I headed off to Berkeley, and trying to advance this, and it's not working. And a girl I met in high school, shown here, named Yvonne Rojas, said something to me that actually stuck with me. And I don't remember the details of the context of the conversation, but she said, genetics is so cool. It sounds so cool. And I knew I was interested in biology. I didn't really know much about genetics. And here we are in Prague, the land of Gregor Mendel. So I got to Berkeley. I was interested in biology, biochemistry, but then I checked out genetics. And I majored in genetics and got my degree there, managed not to get kicked out of school. And at the end of graduation, she was done with college where she went elsewhere. She went to Chicago and she was working at UCLA. And we had both had relationships that had ended and we were both single. And so I said, well, maybe I'll try to look for a job at UCLA. And sorry, this isn't working. There we go. So at that time, I went to UCLA and took a position as a laboratory technician in Irvin Chen's lab. And this was really transformative for me. This was my first real wet lab experience and my first chance to work on a human disease at the molecular level. I don't know if I can get another clicker because I'm pushing this like 6 times. Okay, and so this is a picture of Irvin in his younger days. He's a retrovirologist. He's now director of the UCLA AIDS Institute. And this was really great because I had a chance to meet mentors who would be physician scientists, role models. Dennis Slayman, some of you may know him from his breast cancer work, development of Herceptin. Many of you may know Stephen Neimer from his work on AML. These people were in their 30s just then and just getting started, and many of you probably know John DiPerzio as well from his work on AML. And so this was my first chance to really kind of entertain the idea of combining medicine and science. The very first publication I was on happened to be in science. I didn't play a huge role in it, but this was it. And you can see Dennis there, Joe Rosenblatt, if you know him, William Waxman, and it was just a very transformative period for me. He gave me the opportunity to have my own project, and my very first paper was published in Molecular and Cellular, my first authorship in Molecular and Cellular Biology. I'm glad Jerry didn't dredge that out. I went on to UCLA, and I went to graduate school and medical school, and I worked with Owen Whitty, who made the seminal observation of the BCR-ABL tyrosine kinase, of course, as you all know. And he had also taken in a few physician scientists, including somebody you maybe know well. And he had lowered the bar maybe just enough for Tim to join the lab. And Tim will also note that the discrepancy in head size is actually accurate. It's not really a reflection. It's not a magnification here, for those of you who know Owen. And then Charles Sawyers was also a fellow in the lab at the time. And so I had, again, the opportunity to interact with these incredible physician scientists. And my very first paper to work on CML came out when I was in graduate school, and I was the second author of that PNAS paper that was looking at genomic breakpoints in BCR-ABL positive ALL. And we did southern blots, and I don't want to even go into that technique. And another paper with antiquated techniques was characterization of the BCR-ABL promoter. This was the primary work that I did for my graduate work. And it was back when you could have a figure panel that was just a description of the plasmid you made, as well as CAT assays, S1 nuclease assays. You young people are looking at me like you don't know what I'm talking about, I know. So by this time, I'd been seeing Yvonne, and on the right, we were married in 1991. That was, I think, the smartest decision I've ever made to this day, and I don't know if she feels the same, but I certainly do. So I stayed at UCLA for internship, residency, and fellowship. And at that time, Charles was leading the first in-human study of STI 571, imatinib, and the first cases of imatinib resistance were observed. Now, at this meeting, there's been a lot of talk about resistance, and I think it's important to keep in mind that there's primary resistance versus secondary resistance. And we've heard a lot of talk primarily about primary resistance, which is failure to achieve an initial response, and there are many different unappreciated mechanisms for that. We were more interested in secondary resistance because this is what people were progressing and dying of. This was like the more pressing need at the time, so we wanted to try to understand that. And so Mercedes Gore in Charles' lab, I guess the authors got cut off here a little bit at the end, but Charles was a senior author on this paper, described the very first kinase domain mutation. These were blast phase patients, and this was the T315I mutation. And on the left was evidence of BCR-ABL amplification in a subset of patients. So as Jerry mentioned, we went on to look into a larger cohort, chronic phase, accelerated blast phase, and we found in a very high proportion of patients a number of different mutations, not just the T315I mutation. On the right is a ribbon representation that you all know of the BCR-ABL kinase domain, and showing where these mutations occur by the spheres, by the colored spheres. The red ones are those at contact residues. The rest of them are not at contact residues. And John Kurian's take on this was that those other mutations likely destabilized the specific inactive conformation that's required. On the left, we tried to conceptualize on the cover this idea of pre-existing clones and heterogeneity at the time of resistance, which we had observed, patients with multiple different mutations in different clones. Very nicely, this was dovetailed with work that Sue Branford and Tim had published around the same time. So these were the main findings. This was really the highest level evidence that BCR-ABL kinase activity is critical. I know Rick Van Etten had published, very elegant mouse models, but for human CML, the fact that you could treat with an inhibitor and then get resistance in the target gene really told you that that was primarily mediating it. And then the other couple of things that were important to mention are that a minority of imatinib-resistant mutations occur at contact residues. The majority probably destabilize the inactive conformation, and which makes you start thinking about different ways of binding to the kinase and inhibiting it. But certainly trying to re-inhibit the kinase would hold tremendous therapeutic promise. But getting interest from pharma companies in doing this was really difficult. We first approached Novartis, and there was no interest. There was just this widespread belief that they'd been drinking the Kool-Aid, imatinib was a silver bullet, resistance and tolerance was not much of an issue. Now we have 6 drugs, right? And we're talking about more coming down the pipeline. But Bristol-Myers Squibb had the SRC inhibitor, and because SRC and ABL are so closely related, we postulated that it may develop in a different way than imatinib, since imatinib does not inhibit SRC family kinases. Jerry alluded to this. This was, for me, the greatest day in the lab. The date that I did this experiment is shown on the lower right. That was my wife's birthday, so she now knows what I was doing that day so late in the lab. But what it shows is that the vast majority of these mutations are really highly sensitive, with the exception of the T315I mutation. And this was the first preclinical description of dasatinib. And this is something many of you may have seen, the kinase domain going in motion, and the inactive conformation shown here bound by imatinib in purple, and versus the active conformation shown in blue that's bound by dasatinib. And again, they both interact with the region up here, which I'm not sure if I can't see where that's pointing to. They both interact with the threonine 315 residue. So at that point, I moved up to UCSF, where I've been since then. And we'd gone into clinical trials. We published that you could engender responses with dasatinib, very nice responses, and that the T315I mutation was certainly very resistant. The reason we felt compelled to do it, despite the preclinical data, was that we thought it was unethical to put so much stock in preclinical data when there were patients that had no other options. But we subsequently learned that in addition to the T315I mutation, the F317L mutation was not associated with cytogenetic responses. And this dovetailed actually really well with the relative resistance of the F317L. So the correlation between the preclinical and the clinical was really quite outstanding. This study showed that the responses early on looked very durable for chronic phase, but patients with more advanced phase, unfortunately, did have shorter progression-free survival. And this was, of course, approved in 2006. And I have to say, one of the great clinical moments of my life was actually telling a patient who was on this trial that, not only do we understand the genes that drive your leukemia and we have a drug with imatinib that successfully targets it, but I can tell you with confidence that it's that I know the reason why you've stopped responding to imatinib. I know the specific kinase domain mutation you had, and I've tested in the lab and I know it's sensitive to this drug, and so you're likely to respond. And I realized that in oncology, I was probably the first person to have that conversation. Now that's of course commonplace, and even throughout most of medicine, that was a very, very uncommon conversation. But I actually got chills down my spine when I had the opportunity to have that conversation. We tried to look prospectively to see what mutations in vitro could cause resistance to dasatinib, and a very limited number in vitro could, and these were located at contact residues. And as Jerry had mentioned, we wanted to know what happens to people treated sequentially who develop relapse to the first drug and then to the second drug. And Jerry went over this, so I don't need to go over it again. But we found the mutations that we had identified preclinically and validated them essentially. But the really concerning thing, I'm going to come back to this later, were the compound mutations, because there was really nothing with promise for these. And I think these remain rather scary. And we can talk about efforts to try to prevent them from happening. We also learned from dasatinib that it has a very short half-life. And despite this, it's very active when you give it just once a day. We've also seen this now with asciminib. Once a day seems effective. And we modeled this in the lab, and Corinne Kassip, shown in the lower right, was my first technician hire. She's now on the job market as a MD PhD graduate, HemOnc fellowship graduate. So look out for her. And we also further tried to turn the resistance question on its head and ask, what makes BCR-ABL such a great target? And work that Jennifer Asmussen and Elizabeth Lassiter did actually showed that when you inhibit BCR-ABL, in contrast to inhibiting other mutated kinases like BRAF and melanoma cells, you have MEK-dependent feedback that persists for a long time that seems to prevent these cells from being efficiently rescued. I know, this concept is being investigated by Kendra Sweet through inhibition of JAK for those that are able to escape this. So it'll be interesting to see where that goes. So applying this to FLT3, I don't need to go through this because Jerry, I think, kind of nicely discussed. I just want to mention that Kathy Smith in my lab was my first HemOnc fellow, and she, really laid the framework for and the interest in developing, novel, potent and selective TKIs. And now we have, of course, gilteritinib, quizartinib as the more potent ones. Crenolanib has shown some activity, as Jorge has demonstrated. And and Jerry talked about this much better than I can. What I just want to show you on the right is in gilteritinib-treated patients, you can get extinction of the FLT3-ITD mutant clone. The right one, the upper right one, is actually an NRAS mutant clone, so that can escape. But you can basically see that one of the clones is actually completely gone. So I think it raises the possibility that we might achieve something greater than what we've seen. This is just a list of the mutations that can occur. Notably, 2 patients developed new BCR-ABL mutations on this study as an escape mechanism, which we thought was really kind of jaw-dropping. Okay. Next slide, here we go. So this is, I think, data that we sometimes overlook the importance of. So the hope is, of course, that if you can combine FLT3 TKIs with chemotherapy and AML, you may achieve the sort of improvement in response that we've seen in BCR-ABL positive ALL. So on the left, the blue curve is what was seen. This was a horrible disease. This is pediatric, right? So the majority of these kids were dying due to their leukemia. And on the right, you see what you achieve by adding imatinib or dasatinib to the chemotherapy regimen. And again, the number of patient life years added to this is really amazing. Now, I don't think FLT3 TKIs, because of the heterogeneity issue that Jerry mentioned, Jerry has said it's amazing we cure anybody due to that heterogeneity, and I certainly believe him on that. So in terms of mechanisms, Ariel Leite-Vidal in my lab spoke earlier today. But when it comes to active site inhibitors, the main problem is a loss of affinity for the TKI and not one of ATP affinity. Of course, that can pose resistance to an allosteric inhibitor as well. But what you see for EGFR is you can actually get an increase in ATP affinity with no change in the affinity for the TKI. But this would not be expected to cause resistance to an allosteric inhibitor, as these are non-ATP competitive. But so REL has investigated some mutations that are located nowhere near the binding pocket. As you know, asciminib will sort of orchestrate this kind of self-assembly of the kinase domain in this auto-inhibited conformation shown on the right. That's its mechanism of action. But the resistance hypothesis shown here on the right is that the M244V mutation shifts that equilibrium due to its location. It may preferentially lead to binding of the SH2 domain and activation that way. So REL showed that this mutation is really highly resistant in vitro and likely involves a novel mechanism such as disruption of allostery. She's gone on, as she presented earlier today, to talk about the importance of regions of the SH3 and SH2 domain. Oliver Hanschel and Inga showed very nice similar data. And so what we worry a little bit about in this setting is that these patients may be unrecognized, and moreover, we may see an issue where we're going to see mutations not within coding regions, but maybe in a splice acceptor site for ABL exon A2. A single nucleotide change could destroy that splice acceptor and lead to an A3 fusion. So welcome to the new resistance, as it were. So we, of course, want to try to work towards making resistance futile. So where do we go from here? So the last main thing I want to talk about is best-in-class features. Now, these are all, of course, known to you. I think we'd all agree these are features that we would like all of our TKIs to have. And of these, if we take costs out of the equation, out of the first four, I think we would say both ponatinib and asciminib each achieve 3 out of the 4. Now, is it possible to take the selectivity of asciminib and the imperviousness to mutations of ponatinib and somehow develop a compound that has both those features? And Keyvan Shokat has taken an interesting approach, which is basically to link the two compounds and basically try to tether ponatinib so that it binds selectively to ABL kinase. And in some preclinical data shown here is the inhibition on the x-axis by this links compound compared with the two separate. And you can see that it's ABL1, ABL2 inhibition is essentially equivalent, but most of the rest of the kinases are really spared from this combination approach. You can do similar experiments looking at the sensitivity of cell lines. This is actually not projecting very well, so I'm going to skip over this. But essentially, this looks almost as selective as asciminib in these various cell lines. And if you look at human vascular endothelial cells as a model of toxicity, cardiovascular toxicity, you see on the bottom panel a much greater splay between the T315I expressing K562 cells and these HUVEC cells. But could this be active in some of these dreaded compound mutations? So the one that we chose to look at is this compound E255E T315I, which has been shown to be, by others, to be highly resistant, even to combinations of ponatinib and asciminib. And in some preliminary data, sorry. Next slide, please. Whoops, back. So basically the signaling seems to suggest that yes, you can, in the lower right, selectively shut off this kinase in a manner that's superior to using the two separately. And very early in vivo data show that this, in green, this compound seems to be well tolerated from a weight perspective and more active than ponatinib alone at dealing with this particular mutation. So the last time I attended this meeting before 2009 was in 2008. And the second reason, aside from shifting my research focus a little bit to CML, was in 2008, soon after this photo was taken, we learned that my wife's back pain was due to multiple myeloma. And as you guys probably know, this is a disease that has a life expectancy even today of 6 years. And I spoke to 5 different hematologists, myeloma experts, and I got 10 different opinions on how to proceed. So on the next slide, I came back to this observation that one of my colleagues told me, don't be aggressive, just treat it with one drug after the other. It tends to respond recurrently. But I was really worried about this concept of these compound mutations arising. And I thought, let's just be aggressive early on. And thankfully she signed on, and she's now 10 years post-maintenance therapy and in complete remission for the last 15 years. It's an N of 1, but for me, it's an N of the most important one. And lastly, I just want to close with a couple thoughts about Janet Rowley. I had the opportunity to introduce Kathy Smith, and Kathy was walking around with this expression on her face for a week after having the opportunity to meet Janet Rowley. Janet was really a wonderful, model for women wanting to go into science. And you probably noticed most of the people I showed pictures of in my lab are very accomplished women, and it's fantastic to have them in their contributing. So I want to just close with a couple of words about John Goldman. John, of course, I never worked with him directly, but I got to know him through these meetings. What I was really impressed with about John was his global vision, wanting to expand. I remember Hematology would come up and as he did today, make all these really prescient comments about the rest of the world. These drugs are so expensive. Isn't it a shame that they're not? I was really heartbroken that dasatinib was not available in my ancestral homeland of India for most patients. And so John had that vision, and I really began to realize this at this meeting. I mentioned before the man on the left really helped my grandfather. And at this meeting, I actually met his son, who I had known before, who's a well-known hematologist in Mumbai, and whose first name is actually my given first name. So independently, I became a hematologist. He is still alive, but he is unfortunately too old to travel. John, at the faculty dinner in Oxford 20 years ago, some of you may remember, this is just one last humorous anecdote I'll speak about. He's usually very in command of language, and maybe he had a little bit to drink that night, but he was talking about his undergraduate or his college years at Oxford, and what he meant to say, we realized later, was that Oxford then was all male, whereas now it was for both men and women. But the word he used was, he said, back when I was in school, Oxford was purely homosexual. And we all looked at him in wonderment, like, what could he mean by this, So I don't know if others who were there remember that. But a lot of people to thank listed here, certainly my wife, my parents, the patients who've entrusted their medical care, so we've been able to learn as much as possible. Really, all of you for your contributions. You're all very inspiring to me. I've gone a little bit over time. I apologize, but I thank you. I'm very humbled and honored.
Thanks, Neil. That was outstanding. Do we have any questions people would like to ask Neil?
Maybe it's time for dinner.
Well, if not, I'll ask you. If you had, the NIH just gives you a ton of money and says, Neil, give me the game plan for the next 5 years. What do you think?
For CML or for
anything you want? I'm giving you $5 million. I'm giving you 5 years of funding, man.
I've never even been able to contemplate that. It's so far from my reality. I think there's been people have fallen in love with immunotherapy, but I think that there have been advances in medicinal chemistry, and to where now, we're able to perturb networks that we never were able to do so before. And I just hope that people will remain engaged with targeted therapy. I think people, jumped off the train when they realized that the TKI story from CML was relatively unique. So I would, put it towards, better kind of computational medicinal chemistry efforts to target things that are relatively, undruggable. So Kevan Shokat, of course, who I mentioned, he's developed the initial KRAS G12C inhibitor, and so I think there's so much more to mine there. So that's where I would like to see effort go. I also would like to stay away from immunology, like somebody said earlier today. Apologies.
Amen. Any other questions? We're going once, twice. If not, thank you for a wonderful day, and go look at some posters and see you tomorrow.